Good evening, everyone. Welcome to our fourth live session for this NPTEL course on Introduction to Dynamical Models in Biology. We had a minor technical glitch, that's why we got delayed by a few minutes. Hope you will mind. I welcome you all here in this live session. As we have been done in earlier cases, we will discuss three things. First, we will discuss about the key point, the thrust areas for this week's lecture, the points that you should focus while listening to these lectures. At the same time, the second thing that we will do, I have some practice problems which are not part of the assignment but related to this week's course. So I will try to discuss this with you and try to solve them in front of you here in the live session so that you get an idea how actually you are supposed to solve these problems, right? And lastly, I will pick up in between, obviously at the end also in between, if you have any queries, you are free to ask the question, write that in the chat box so that I can respond to them. Please don't hesitate that you don't have to wait the whole live session to ask your question. At any moment of time, please post your question so I will try to answer that question, right? And you can, the focus would be this week course only, this week lectures only, but if you have any query from previous one also, you can ask the question, right? So let's start the live session. Few people have already joined. I hope others will also join with time. Let me share my screen so that I can discuss the key points of these week lectures, right? So hope you people can see my screen. See, in this week's lecture, we are focused on the dynamical properties of certain network motif. If you listen to the lecture in the third week, in one of those lectures, we have discussed about network motif. There are many types of network motif. For example, a negative feedback, right? Negative feedback can be a network motif. For example, I have supposed A, B, and C. A activate B, B activate C, and C inhibit A. So this is a negative feedback. In this way, we have positive feedback. We have also incoherent feed forward, coherent feed forward. All these type of network motifs are there. And in this week's lecture, what we are doing, we are going to discuss. We are taking some simple network motif and try to understand some characteristic behavior, characteristic dynamics of these motifs. What do I mean by characteristic behavior? So this is an important word, characteristic dynamics, right? In textbook, in lecture, people will say this motif has this characteristic dynamics. By characteristic dynamics means, suppose I take a network motif, right, and I give an input which is obviously time dependent, right? So time versus something like this, and I get an output. So suppose that one is something like this. This is also time dependent. So if I have a particular motif has a particular type of input-output behavior, that is called the characteristic dynamics of a particular motif, right? So in this week's lecture, this fourth week lecture, we are actually discussing characteristic or unique dynamical property of negative feedback, positive feedback, incoherent feed forward, right? And what you should focus on? You should focus on certain points regarding these motifs. For example, negative feedback, you will realize when you listen to this lecture, it does not matter. For example, in my lectures, I may have discussed about a genetic circuit which is made up of negative feedbacks. Or suppose I have discussed a positive feedback using enzymatic reactions like the cell signaling pathway. But that does not mean it is only the property that we will discuss, the unique dynamical property that we are discussing is related to transcription or cell signaling or enzymatic reaction like that, right? We are picking up gene expression, enzymatic reaction as an example and writing the ODE. But if the parameters and the functional structure is same, a negative feedback in one system can have similar behavior in negative feedback in another system. That we have to keep in mind. For example, one unique property of a negative feedback, a unique property of negative feedback is to give monostability. That means the system will have only one stable steady state. What does that mean? That means wherever you start, you take a two-dimensional system. Let me explain here. Taking a two-dimensional system. Suppose I have two-dimensional system, two variable x and y, both are changing with time. So then what will happen? And suppose here I have one stable steady state. So wherever you start, any of these places, if you start with this cross, eventually it will collapse here in the stable steady state. If you start in this square box, eventually the system will collapse here. I am just drawing this curve arbitrarily to show that in the phase space, wherever you start at t equal to 0 initial time, the system will reach a monostable condition, that particular stable steady state. Now this has an advantage. Suppose my metabolic system is in monostable system right now. Suppose, then what will happen if there is slight fluctuation because something else I have eaten which I never used to eat, or suppose slightly there is change in temperature or some hormonal activity in my body has changed, those perturbations will not affect the metabolism as a whole too much if it has very good negative feedback and the system is monostable. What will happen? My metabolism will come back again to the same stable steady state or near it. That means it is resistant to some extent to perturbation, right? These property you have to understand. But will it happen? Always no. Obviously, your negative feedback, the functions, the parameters, the structure of the system has to match. But if it's a negative feedback with right parameters, then it can give rise to monostability, and most of the time it gives rise to monostability. What usually is not so common, but negative feedback is the circuit which can give rise to is oscillation. Only negative feedback can give rise to oscillation. Not always, but it can give rise to oscillation. Let me discuss when it can give oscillation, and the repressolator that I have discussed in one lecture is actually a negative feedback that gives oscillation. In the web page, I have uploaded also a few paper, given link for few paper where this original repressolator was discussed. Please go through them. You may not understand all the all the experimental condition, but try to grasp the idea what is happening. A repressolator is something like this: A inhibit expression of B, B inhibit expression of C, C inhibits expression of A. So this is minus, this is minus, this is minus. So I have 3 minus. So that means the end result is also minus. So it's a negative feedback. And it gives rise to oscillation if particular parameter value matches. Not always. That is important. If the parameter's value does not match, it will not give rise to oscillation. It will give rise to monostability, because monostability is a property, a characteristic property of a negative feedback, right? We have discussed separately that in a negative feedback circuit in a particular lecture in this week. So you should check that. Now, one important point: a negative feedback will give rise to oscillation only when there is a delay. So a delay in a negative feedback can give rise to oscillation. So delay is another requirement. First requirement, it has to be negative feedback. Second thing is it has to have delay. What do I mean by delay? Delay means there one process start and the second process start. Before these two, there is a time gap. So that is why if I take a negative feedback, A activate B, B inhibit A, either enzymatic pathway or transcriptional circuit. A is a transcription factor, B is a transcription factor. A activates B, B inhibits A. This will not give rise to oscillation. This is negative feedback, but it will not give oscillation, because there is very little delay between these two processes, one activation and another inhibition. Whereas this repressolator, which has 3 molecules, so this intermediate B is actually causing delay in signal transfer from A to C. So that's why this gives rise to oscillation. So somehow, either by a molecule in between, one or two molecule in between in A and C, you can give rise to delay. Or sometimes what happens, molecule has to move, right? Molecule from cytoplasm has to go into nucleus, from Golgi it has to come out in the cytoplasm. All these phenomena happen. So these are diffusion process. So diffusion can be slow, chemical reaction can be fast, right? So in that case, diffusion can add delay, and the negative feedback can give rise to oscillation. So what I am trying to tell you is that we have two lectures. Both of them are negative feedback. One shows the negative feedback is giving rise to monostability. The other one shows that the negative feedback is giving to oscillation. You have to understand a negative feedback gives oscillation only when there is a time delay between processes, right? So that's why I have picked up the example of refrigerator. Now, beyond this negative feedback, we have discussed also about positive feedback. And positive feedback has a very characteristic behavior which is very useful in biology, which is just opposite of negative feedback. What I said, if I have a good negative feedback in my metabolic circuit, metabolic pathways, right? Then if there is perturbation, my metabolism will come back, right? That's what will happen. Now, positive feedback does the just opposite. So what is a positive feedback will do? A positive feedback amplifies perturbation. So disturbances will be amplified, right? So look into the example of positive feedback that I have discussed. In that case, perturbation, amplification of perturbation is not discussed in so much, because that requires to some extent, stochastic analysis of the model. We have not discussed that. We are discussing a deterministic one. But what this thing leads to is that a positive feedback gives rise to bistability or multi-stability, right? If you remember multi-stability, bistability means two stable steady states. Multi-stable means more than two, obviously, right? So it is just opposite of monostable. Your negative feedback circuit will make the system monostable, whereas a bistable or a positive feedback will break it away from monostable, make it bistable or multistable. And as we have discussed during bifurcation, these multiple stable steady state give rise to heterogeneity in cell. One cell type can become two different cell types. Cellular differentiation can happen. So positive feedback plays a role there. Again, you have to remember all positive feedback will not give rise to bistability. The parameter and the mechanism for which so the function that we are using in ODE should be proper so that we have bistability. Is there any question till now from the audience? If anybody has any question, you can post it. Okay. If there is no more question, let me go and discuss the last important point in motives is the incoherent feed-forward. Incoherent feed-forward is a motive which gives rise to adaptive behavior, right? So what is that? Incoherent feed-forward give rise to adaptive behavior. So what you have to understand in this lecture, there is a lecture on adaptive behavior using incoherent feed-forward. What you have to understand? What is adaptive behavior? That you have to understand clearly. And then you have to understand the structure of incoherent feed-forward, the property of that topology, the structure which gives rise to this adaptive behavior. And then focus on the equations and how we are analyzing to show that this motive is adaptive, that it is insensitive to the external signal. Or whatever be the signal value, the system comes back to the same stable steady state, right? That you have to understand. So that if I give a motive or if I give you a set of ODE, you should be able to check whether that has adaptive behavior or not. Fine. So these are the points that you have to focus on while listening to watching the lectures for this week. The last lecture, the sixth lecture, is on databases and web resources. Some of the databases which are used for many of our work, for example, cake database or the CellML database for the models, all these things that I have listed there. Remember, that is not an exhaustive one. It is not an exhaustive database listing. It's an example, so that you get interested to look more into it. So that is what we discussed in this fourth week, on the last week of our this course. One thing, if you are serious about learning dynamical models, I will tell you one thing that we have not discussed, but important to some extent for dynamical models in biology, that is called parameter estimation. Right? We have not included this in the course because that will require some more complicated mathematics, and also it will lengthen the course. As it is an elementary course, our intention is to make you interested, inclined toward learning and using mathematical models. So we want that you start using ready-made model, you borrow from others, borrow parameters from other people's model and use it. Once you get a hunch of the system, when you get habituated with building models and analyzing them, then you should go into parameter estimation thing, right? There are texts available, but we have skipped that one. But remember, parameter estimation is not a trivial thing. There are multiple methods, but none of them is a foolproof one. So people do trial and error, and multiple difference of opinion exists. But this is one area which is important, but we have left that one because this is the elementary course. So this is from my side on what you should focus on this week's lecture. If you have any query, please post the question. I will wait for 1 minute. Sriram, Bharat, all the few people have any query. If you are still online, if you have any query, you can ask the question. Okay, it seems nobody has any question right now. So let me pick up a problem for this week's lecture. It's related to this week's lecture. Hope you can see the question on the screen. The question says, there are two proteins and they control each other expression. As simple as that. The dynamics is represented by a OD. I have given you the OD. You can see the OD written here. 2 OD, X for dx dt and dy dt. The question is, from looking at the checking the OD, you have to tell what type of regulation. By what type we mean the negative feedback, positive feedback, coherent feed forward, some bifan or something like that, right? So you have to tell that, and you have to calculate how many steady state do you expect for this system. So let us first try to answer what type of regulation is this. So will someone try to answer this? If anybody who is online will try to answer this question. Check the ODE. Dx dt is equal to 1 divided by 2 plus y minus x, and the second ODE is dy dt is equal to 1 by 2 plus x minus 1. So what type of regulation is this? What type of motif I should draw? So for example, I draw x here and y here. I can use arrow, I can use hammerhead. So what type of diagram it should be? Positive feedback, negative feedback, direct one-way relation, whatever it is. Tell me. Any guess? Sriram, you have said negative feedback. Yes, it is negative feedback. Is it negative feedback? Can you think? Okay. Can you just write what is in one line why you think it is negative feedback? Okay, Sriram may be typing. Let me look into it. There is negative. Sriram, you are right. There is something negative in it. Why it is negative? Look at the first term. Suppose for dy dt, you look in this term. This is for the production of y, right? Yes, change in x inversely proportional to y. Absolutely, you got the logic. You got the logic, but you slightly missed the point. It will not be negative feedback. There is negation. What is happening? Just let me draw this x versus 1 by 2 plus x, right? This 1 by 2 plus x is in the second OD, right? So if I increase x, what will happen? Initially when x is 0, this value is 2 somewhere here. X is 0, this is 2. As x increase, as Sriram has said, this 1 by 2 plus x will decrease, right? So it is decreasing. So that means as x increase, y decrease. So it is not a hill function, but it is similar to that. It is similar to that, isn't it? So it is in decreasing. It will not be a rectangular hyperbola, just drawing arbitrarily that. So that means x is inhibiting y, right? Now you do the first OD, that also a similar thing. The only thing is now it is for x and you have 1 divided by 2 plus 1. That means this case y inhibit x. So now you have 2 negation. So Sriram, will you still call it negative feedback? Negative feedback is when I have a molecule which tells B you activate and B says back to A you get inhibited. This is negative feedback, right? The one x inhibit y inhibit x is not negative feedback. This is mutual repression or inhibition, right? And in fact, this will work as a positive feedback. Why? Because this is negative and this is negative. Negation of negation, negation of negation is positive, assertive, right? So if I have minus into minus, I will get plus. So what is happening in this case? X is telling to y, do not do. Y is saying x, you do not say it. So then as a whole, y is getting option to work, right? So negation of negation, positive thing is, right? So this is a mutual repression. Hope it is clear. So you have to be careful. You rightly pointed out there is negation, but I have 2 negations, not 1 negation, right? Negative feedback will have eventually only the net. If I have a cycle, if I have arrows and if I multiply the sign of each of those arrows, eventually the net result should be a negative, right? In this case, this is a positive. Okay. Now let us go to the second question. How many steady state do I expect in this case? How many steady state do I expect in this case, right? How do I do? Should I go for detailed analysis? Yes, I can do detailed analysis, solve this equation, right? But I will not do those. I am a bit lazy. What I will do, I will use shortcut. Bharat says I got 2 steady state, but the value of x for the steady state is coming out to be irrational number. What do you mean by irrational number? What type of number you are getting, Bharat? So let me write down. By that time, Bharat will type. I will read out. Dx dt is equal to 1 plus 2 plus x minus y minus x, and the other one is dy dt is equal to 1 by 2 plus x minus y. As I said, I am lazy. I will not do all hard work of the. I think Bharat is solving the equation. I will not go into those. I will not take those trials, right? Maybe in the final exam, it will be given 1 mark or 2 mark. Why should I put so much of effort? I mean, don't follow my this idea, but I'm just giving here. you can do detailed analysis. I will use null clients, right? Square root of 13 divided by 2. Why do you think it is irrational in that sense? Okay, you get a number. So let us see, right, how I will do. The question says how many steady state we can have. So I will calculate the null climb. So x null climb. Dx dt equal to 0. So that means I get x equal to 1 by 2 plus 1. Simple. You take this equation, put left hand side 0, separate out x and y. As simple as that. Okay, y null climb. dy dt equal to 0. So put 0 here. dy dt equal to 0. So what do I get? 1 by 2 plus x minus y equal to 0. So that means I will get y equal to 1 by 2 plus x. Now one thumb rule. When you draw null lines on a paper and pen, you should always use different colors pen. This is x, this is y. Remember, x and y are two genes. So that means I will deal with only on the positive quadrant. I will not look into what is happening in the negative quadrant. Okay. First, the y null line. It is easy to draw. Just now, few minutes back, I have drawn it. Okay. So that should be y equal to 1 divided by 2 plus x. Put x equal to 0. We call it flagging. You know, the extremum checking. Put some value of variable to 0, 10, 100, infinity, and see how the function behaves. This is a thumb rule how you should check. If you do not have a plotter with you, you are supposed to check this way. If you are on a desktop, why don't you open Google and plot the equation, or Desmos and plot the equation? You can see. Otherwise, you can follow me. So I'll put x equal to 0. If x equal to 0, y will be half. Right? If x equal to 0, y is half somewhere here. I think previously I have drawn this one few minutes back. I said it will be 2, but it will be half, right? Half will be correct. Fine. And then, as x increase, y should decrease. And it should decrease almost like not exponentially, but something like this. And I will get an asymptote. Remember, I have an asymptote here. So this is my, this is my y null. Now I will draw the x null line. What is my function? X null line is this one. This is symmetric, right? Now in that case, just it will be opposite one. So it will start from half, and in the y side I will get an asymptote, right? So somewhere here maybe half. So it will start from here and go roughly something like this. If you have a plotter online, Desmos or Google plotter, try this and hopefully you get the same thing better than my drawing. This is my intersection point. That's why I insist use two different color, blue and red. I can easily see the intersection. This is my steady state. Now looking at it, I can see that whatever parameter you do, there is no option that these two null lines will intersect more than 1. So I will always get only one steady state. Right? What is the numerical value of it? Okay, for that you have to solve it. Right? The question has not asked it, so I will not take the trouble of solving it. In general, this is the stable, this is a single steady state. You can, if you have online, fetch plane plotter is open with you. Earlier I have given a link for fetch plane plotter. You can plot the fetch plane, right? Fetch portrait of it. You will see possibly this is a stable node, right? Everything will sync there. Fine. Is it clear, Bharat, now? Any doubt? Okay, so hope it is clear to everyone. I'll go to next problem. Bit lengthy problem. Try to follow. Don't get confused. From childhood, from school days, I have followed one principle I have learned as a student. If the question is long, the answer will be short. Right? So the examiner is trying to actually confuse you or trying to save his or her skin by giving details, either of it. So here what is said? Expression of a protein is induced by an external signal S. S is an external signal. The protein controls its own expression by a negative feedback. This is another control. S is one external signal that controls the protein expression, and what do you say? The protein itself controls its own expression by a negative. Now it is said that these two controls are independent. Remember, this is very important because many a time the two transcription factor works together at the promoter. Then they are not independent. Both of them are required in the right concentration to have induction or inhibition. In this case, it is said they are independent. So your life is easy. And what is said? That both of them follow some sort of Hill function. Obviously, one is negative, one is positive. So there must be an inverse and normal Hill function. The Hill constant is 1 and Hill coefficient is 1. Here the examiner is making your life easy by making them 1 so that your calculation and plotting will be easier. Now, what the question says, create an ODE based model. So you have to write down the ODEs and calculate the steady state when the signal is 1. That's what we have to do. And you have to consider that there is no leaky expression. Again, the ODE will be simple. And I may consider all rate constant equal to 1. Just please read it. I have read this one for you, but please read yourself the question once. Take 1 minute to read. Then I will start it. Try to understand. As you read, I will read out again. This protein is controlled by two signals. One is an external signal, S, which increases the expression. Whereas the protein itself gives a negative feedback. Right? And both of them are independent of each other, and both of them you can use the model using Hill function, some sort of Hill function with Hill constant 1 and Hill coefficient 1. There is no leaky expression. You have to create a ODE based model and you have to calculate its steady state when the external signal is 1. Fine. So how should I cross it? All this problem becomes much easier if I draw it, right? So let me draw it. Small p is the protein, so G is the gene, right? It gives rise to mRNA, right? mRNA get degraded. These give rise to protein P. Protein also get degraded. I have a signal S which positively regulate, and I have this one coming and negatively regulate. Right? So this is negatively. This is the structure. If you have followed my lecture earlier, sometimes transcription translation we club them together. We do that with the assumption that the mRNA has reached steady state very fast with respect to protein, and we do that when we have lots of things to model, so you want to reduce the number of OD. In this case, I do not need that. I will have just 2 OD. Why should I bother to club transcription and translation together? I will keep them separate, right? So let me write down the ODs. So DMDT for mRNA, what I will write? The first thing is S is positively inducing and with a Hill function. It is said here, right? It follows Hill function with constant 1 and coefficient 1. So I will write something like this. Hill function I will write. Okay, I need a constant here before that, and as the question said other rate constant equal to 1, so I have considered that 1 and I am not writing that. So yes, Hill constant coefficient is 1. Hill constant it is said 1 plus s to the power 1. I have one rate constant here plus another rate constant 1 into now negative control by the protein itself. So it will be 1 1 plus protein to the power 1. Again, it is a Hill function. This is also a Hill function, but inverse Hill function. Minus degradation of the mRNA. These two terms are the production. Now, degradation of the mRNA. Okay, I will again consider, as the question said, I will consider 1 as the rate constant for degradation and the concentration of mRNA. So my life is easy. Lots of 1. S, P. So let me write it down cleanly. I will have S by 1 plus S plus 1 by 1 plus P minus M. Simple. So what will be my dPdt for protein? Again, rate constant is 1 for translation into mRNA concentration minus, again a rate constant 1 I have considered because the question has said, otherwise I have to take another value, 1 into degradation, so concentration of the protein, this is M minus P, right? So the first part of the question is over. It has said create a ODE based model. This is my ODE based model, right? In our assignment and the final exam, we are not doing a paper and pen exam. So this type of question will not be asked that you have to write down the ODE and do that. But something opposite can ask, right? I can give you a model and then say, okay, tell me which of the diagram is correct for this model, which of these network models is correct for it, right? Something like can be done. That's the inverse one, right? So get prepared for it so that you write down the equation, so that looking at the equation you can get back to the diagram, right? So now this is my model. Now comes the second part. Before I go into second part, does anybody has any question here? I hope if you are following, then you should be able to write down simple model like this for yourself also. Okay, so if there is no question, let me move to the second part of the question. I have to calculate the steady state value for, I have to calculate the steady state for S equal to 1, right? So that means, let me write down the equation. dm dt will be S is 1 divided by 1 plus 1 by P minus M. The other one is dP dt equal to M minus P. So the second equation is the easiest one, M minus P. Start with that. Use for steady state. What you will do? M equal to P, right? Because dP dt is equal to 0. So I have got M equal to P. Now I will go to the first equation. What do I get? dm dt equal to 0 is equal to 1 by 1 plus 1 is 2 plus 1 by 1 plus P minus M. Now I will replace P by M. So it will become 2 plus 1 plus M, because M and P are equal in steady state. I have got from the second equation minus M. So someone would try to solve it. What will be the value of M? Sriram, I will come to your question. Please hold on. Let me finish this part. What will be the value of M? Can someone try? What will be the value of M solving this? Again, I will be lazy. Actually, looking at the equation, one can say what will be the value of M. You can make a quadratic equation out of it and solve it, but even without that, you can say what will be the value of M. Remember, the value of M has to be greater or equal to 0. We cannot take a negative one, because M and P are positive things, genes and proteins, right? mRNA and protein. So they cannot be, concentration cannot be negative. Any try? What is the value of M? Okay, I don't know whether anybody is trying it or not, but looking at that, you can easily say consider m equal to 1, you put it half plus 1 by 1 plus 1 is half minus 1, so you have 2 half, 2 half is 1 minus 1 is 0, right? So that is m equal to 0, so that's 1. So the steady state value is m equal to 1, p equal to 1, right? So again I will suggest you, I had earlier shared a link in the course page for drawing the phase portrait, right? So why don't you try to plug this equation, this one and this one, and draw the phase portrait and see what type of steady state is this. P equal to 1, M equal to 1. Is it stable, unstable, spiral, whatever it is, right? The center type, just check it, right? So that will give you a better idea about this system. Now I will come back to Sriram to your question. I will write down the equation that I have written. What Sriram is asking is the Hill equation only for signals. Isn't protein acting as an inhibitory signal? Yes, the protein is acting as an inhibitory signal. And the question has said, the question examiner has said that for both the control by external signal S and for the protein, for both you can use Hill function, right? So what I am saying is that I have this gene. So mRNA is produced. So this is my thing, right? So the protein somewhere here is giving a negative control, so negative, and the signal is giving positive, right? For both I am allowed to use Hill function. So what I use for the first the signal, external signal m, dm dt is equal to, I have considered s by 1 plus s. So this is the Hill function, standard Hill function, because if you remember, Hill function is y equal to x to the power n, k to the power n by x to the power n. N is the Hill coefficient, k is the Hill constant. The question has said n equal to 1 and k equal to 1. The question has said, asked me to consider those values. So I have written these. So this is a Hill function, standard Hill function, right? So this is positive. As S will increase, the induction will be more. In this case, actually, it will be hyperbolic, not this sigma, right? Now comes the next part, inhibition. And that's why what I have considered again a Hill function, but inverse Hill function. What is inverse Hill function? That will be k to the power n divided by k to the power n plus x to the power n, right? So in this case, k is 1, n is 1. So it is 1 divided by 1 plus p. So this is inverse Hill function, right? Minus the degradation m. Fine. So Sriram, can you see now that both the signal, there is signal coming from the outside as well as the signal given by the protein itself, both are included, right, in my OD, right? One thing is that I have used a plus sign here. This is important because these two S and P control, this positive and negative control, are independent. That is said in the question paper. If I go up in the original question paper, and if you remember, two controls are independent. How I am maintaining that independence? See, I have a plus sign in between. So when S is 0, the effect of P does not get negative. It is still there. When P is 0, then also if there is no effect on the effect of S. So they are acting independent, right? So that's why I have a plus sign. If they had dependence, I possibly would have used another function or a multiplication, right? So is it clear up to this? Is it clear? Okay, is there any other questions? Yes, Bharat? Yes, Bharat, please go ahead and ask. I'll wait for a few minutes. We are almost at the fag end of our live session. We have 5-6 minutes. Slide with derivation of n for this question. This one you are talking of? You mean how I have calculated the steady state value of n? Okay, so what is the question? Okay, fine, no problem. So my calculation is right, hopefully, right? Okay. Fine. So, is any other questions are there? I'll give you a tip whenever you are creating a model, right? See these parameters and the more and more complicated function we write, many times they become difficult to analyze. So the thumb rule of all these things is always try to put values of 1, 10, 100, something like that, preferably 1 whenever possible, and whenever possible 0. As long as the meaning does not get lost. Then what happens, your equations become much more much more easier to handle. And once the equations are very easy, then I will actually not try to solve it, never. What I personally feel, it may vary with individual to individual, I always feel it more comfortable when I can visualize. And thankfully the web-based online tools for visualization of function is a great thing. So what I will always do, I will simply put them on a plotter and plot the equation and see how they are behaving. Once you plot this behavior, then you think, okay, I have considered one parameter as 1. Okay, if I make it 3, how it will happen? What will happen? So you change the parameter and you see what is happening. So when you are doing this analysis graphically, even if it's a complicated problem, if you are plotting it and graphically visualizing it, then it becomes very easy to understand the dynamics of the system. Then I always try to solve the equation if required manually or by using a computer or something. Because then if I do some mistake somewhere, even if I am using a computer, a MATLAB code to solve the equation, many a times, or JSIM, many a times we can do mistakes. Somewhere one semicolon is missed or one multiplication sign is replaced by some other symbol, your whole answer will come completely different. Right? In a large code, you may not detect it also. Now, if you have done graphical analysis somewhere beforehand, you know how the function should behave. You know the ballpark, you know the goalpost, what can happen, what cannot happen. Then if you get a very strange answer, right? So then in that case, what happens is that you can immediately get and correct it, right? No, Sriram, unfortunately, we cannot use online plotter, right? You will have on the standard what we say, the calculator that you get for online exam in JEE and get all these things, you will get that one. You will not have online plotter, but it's a habit. It's a habit. For example, I keep Desmos on my desktops, what's a bookmark, right, or Google Plotter. Whenever I think something, I simply plot it. So now what has happened over the year, as I kept on plotting, right? When I see an equation, I can visualize it also. So if you also do it regularly, couple of weeks, if you are preparing for the exam of this course, I am sure you will get habituated. And most of the function that you will dealing here in this course, you will remember the function's behavior, right? So plot them. You have not to go and plot some exotic function, right? You don't need. Just look into the functions we are using in all these models and plot them and change the variables and see how they look like. And once you do or twice you do it, it will be in your mind and you will not need a plotter there in the exam, right? Any questions? We have just 1 minute left. Okay, if there is no more question, please remember the 4th week assignment is on, right? So please submit in time. Still, if you all the course will be over by this week, if you still have any query, please post it, right, on the what we say the this discussion forum, right. I'll try to clarify. After the assignment due deadline is over, I'll also post the as a solution. Even if you have any query, then you can also post the queries. And if you are finding that I'm not responding to those queries there, you are always free to write to my official email ID, which you can easily get from the website, right? Write to me if you have any query. Till the exam date, if you have any query, please write me back saying you are a student of this course and you have this query. Don't hesitate. Thank you. Thank you for Raju has do any or two more curves even occur to have different functions? Do any two or more curves ever occur to have different functions? Now here functions, what do you mean by functions is not clear to me because we are plotting functions, right? Yeah, it can have two functions can have same shape. For example, for sigmoid type of shape, you can have lots of. I can imagine immediately 5-6 different types of function which will give the same sigmoid behavior, right? So it can happen. So that is true. It can happen. Okay, so we are done. We are already 5:13 now. So I hope I have been able to clear doubts. And this is the last live session for this course. I hope you have learned something and will continue to learn. Thank you for joining for this live session. Please submit the assignment. Please attend the final exam. And I hope you best of luck for your final exam and all the assignments. And happy learning. Thank you for joining us today.